Hi, my name is DELGEME. I am from Mali, and I'm a postdoctoral fellow from the DELGEME program. I'm based at the Malaria Research and Training Center in Mali at the University of Bamako. I first earned a PhD degree at the School of Pharmacy of Mali, and I was working on the human factors that were implicated in the clearance of chloroquine-resistant Plasmodium falciparum parasites. And then I moved on to the University of Pennsylvania in the United States to get a PhD degree in cell biology. And from then I moved back to Mali, where I'm actually doing my postdoctoral training at the MRTC. So when I was at the University of Pennsylvania, my doctoral training was on studying one parasite that is closely related to malaria that we call Toxoplasma gondii, and it was about learning how those parasites multiply. Now at the MRTC, I hope at the end of this training to be able to focus my work on the cell biology of Plasmodium parasites. So the community has made a lot of progress in the fight against malaria, but we have still not been able to eradicate this disease. And it has been recognized that malaria transmission from the human host to the mosquitoes vector is essential if we want to eliminate malaria. And there is an increased need to develop new anti-malarial drugs and also vaccines to reduce malaria transmission in endemic areas. So my project will focus on the transmission of the parasite from the human host to the mosquito vector. So the parasite is really smart, and he has found ways to have multiple forms within each one of the hosts that he usually infects. And there is one particular one that the mosquito will take during his blood meal that we call gametocytes. And so what I want to study is how the gametocytes move from the human host to the mosquito vectors, and how those gametocytes are able to produce what will be injected in another human being and continue the cycle of the parasite. I will use the bioinformatic tools and the next generation sequencing technologies to identify marker or signature that are associated with the ability of those forms of the parasites that are transmitted to the mosquitoes' vectors. And if I am successful, the study of the Plasmodium falciparum vectors associated with malaria transmission from human to mosquitoes hosts in our natural environment will generate new markers and targets for anti-malarial drug vaccine development.
Hi, my name is Tobias Apinjo. I am a DELGEME Post-Doc from the University of Buea in Cameroon. We know that the differences in parasite strains and the way they are transmitted are necessary if we want to develop and improve elimination strategies. So my project seeks to use advanced tools to assess how parasites from forests and grassland areas of Cameroon collected at different time periods, especially following the implementation of control strategies, will differ. This information would provide useful data that would be relevant for the trials of future drugs or vaccines.
Hi, my name is Enio Cheryl Oriero, a postdoc fellow on the DELGEME program. I'm from Delta State, Nigeria, and my background is in biochemistry and molecular biology. I obtained my PhD from the University of Antwerp in Belgium, and my PhD project was focused on developing new molecular tools for field diagnosis of low-density malaria infections within the context of malaria elimination. There are 5 different parasite species that infect humans, and so far all the tools that are being used for control and elimination of malaria target the most predominant species. The effect that these tools have on the other parasite species is not known, and therefore the focus of my project is to use new technologies like next generation sequencing and bioinformatics to study one of the other parasite species. I would like to see how this parasite species is different from the other parasite species, and especially if the drugs that we're using for control and elimination of malaria are having any effect on this parasite species. Results of my research will be useful for malaria elimination campaigns and will provide more information on how to handle this particular parasite.
Hi, my name is Charles Arama. I'm from the Malaria Research and Training Center at the University of Bamako. I'm a DELGEME Post-Doc Fellow. I hold a PhD degree from School of Pharmacy of Mali, studying host immunology, immunogenetic factor involved in susceptibility to malaria in sympathetic ethnic groups. I then obtained a PhD in immunology from the University of Stockholm, Sweden. From my doctoral training, I work on the topic of novel immunization strategies and inter-ethnic differences in response to malaria infection. Now at MRTC, I intend to set up a laboratory focusing on host epigenetic and cellular immunology against parasitic diseases. Concerning my project, individuals living in area of intensive malaria parasite transmission develop resistance to malaria diseases after repeated exposure to the parasite. This resistance involves many factors that are not well understood. My project focuses on the study of host epigenetic factors and protection against malaria in sympathetic ethnic group of Mali. The project aims to investigate whether those factor characteristics are of importance in differential susceptibility to human malaria infection. Using bioinformatics tools, we want to understand and identify genetic biomarkers of protection against malaria.
Hi, my name is Mireille Amia. I am from Côte d'Ivoire, a country of West Africa. I am a PhD student at Félix Houphouët-Boigny University of Abidjan and fellow of the DELGEME program. We know that Fulani people who live in countries like Mali don't get infected by malaria like the neighboring tribes. Studying the factor responsible of its malaria protection could be a great help in malaria vaccine development. In this study, we are focused on Fulani from Côte d'Ivoire. The main aim of this project is to study the resistant pattern of the Fulani community in Côte d'Ivoire in comparison with the Lobi ethnic group, using new technology like bioinformatics. Results of this project will help to understand malaria immunity of this Fulani ethnic group and contribute to the development of malaria vaccine.
Hi, my name is Tata Roland Bantar, a DELGEME PhD fellow from the University of Buea, Cameroon. My master's research work was on the diversity of the main malaria-causing parasites in Africa along the slopes of Mount Cameroon. Since the stage of the malaria-causing parasite that is transmitted to mosquitoes and later to humans is not directly responsible for signs and symptoms of the disease, not much emphasis has been laid on it in the past. However, for malaria elimination to be achieved, this stage must be well studied. My project is therefore based on exploring why in some populations of parasites this stage is more produced, hence leading to more transmission than in other parasite populations. We will also look at how this stage of the parasites differ in areas of low and high malaria transmission. The results from this project will help in future designing of tools to target malaria transmission and hence elimination.
Hi, my name is Awa Koulibaly from Mali and I'm a PhD student in bioinformatics at the University of Cape Town under the fellowship of DELGEME. I currently work as a bioinformatics consultant in the support of the ACE Mali, which is the African Center of Excellence in Bioinformatics program, and International Center of Excellence in Research in Mali. The ACE program is run with the guidance of NIAID, OCICB, Bioinformatics and Computational Biosciences branch. Despite numerous efforts to combat malaria, the disease is still affecting millions of people. For the last 10 years, artemisinin-based combination therapies has been used as the best treatment for malaria. Although this drug is still efficacious in Mali, our drug efficacy studies have shown that in some regions of Mali, parasites are eliminated faster than other regions. So we suspect that this might be due to the parasite genetics difference. So the goal of my project is to identify this difference using bioinformatics technologies. Understanding the genetics of the parasite resistant to artemisinin will be important to monitor the emergence and the spread of the resistant strain. So to help, and also to help the Health Ministry to determine treatment based on location.
Hi, my name is Bakari Fofana. I'm a medical doctor and also I'm a PhD fellow for DELGEME. I earned my MD degree in 2002 on Plasmodium falciparum genetic markers involved in resistance to sulfadoxine and pyrimethamine. Also, I earned a master in clinical research at KCMC from the Tumaini University. And actually, I'm working as PhD student in genetic factors of Plasmodium falciparum involved in incidence density of clinical malaria in Africa. So in a given area, several factors could affect the number of malaria cases and variation in genetic composition of Plasmodium falciparum population could provide additional information that can lead and facilitate the understanding of such changes and could support science-based decision-making process regarding to malaria control and elimination policies. So we demonstrated that some children experience up to 8 or more episodes of malaria while others just experience only 1 to 3 episodes of malaria during 2 years of follow-up. So the main objective of my proposal in this PhD program is to investigate the implication of parasite genetic profile and on the density, incidence density of clinical malaria in Africa, particularly in Mali. So we will be using the next generation sequencing on Plasmodium DNA using dry blood spot. These dry blood spots were collected in malaria patients from endemic site of Bugula Hamo to determine the genetic relationship between genetic differences and genetic of subsequent parasite found during consecutive episodes of malaria.
Hi, my name is Lona Jemosop Chebon. I'm a PhD DELGEME fellow and I currently work in the Malaria Drug Resistance Laboratory of the US Medical Research Directorate in Kenya. I earned my first degree in biomedical science. Then I proceeded to do my master's degree in molecular medicine in the Institute of Tropical Medicine and Infectious Diseases still in Kenya. Now my master's work focused on looking at the parasite genetics that influence phenotypic trait in the Western Plasmodium falciparum parasites. This was important in studying the parasite genetic diversity, which was important in monitoring susceptibility of the parasite to artemisinin combination therapies, which are the first-line treatment mostly used here in Kenya and also in sub-Saharan Africa. Artemisinin combination therapy is a first-line treatment that is used here in Kenya and most parts of sub-Saharan Africa. And the problem is that artemisinin resistance has emerged in Southeastern Asia. Now, As in Africa, since we use artemisinin combination therapy as the first line, we are concerned about the spread to Africa, and therefore my study is important in focusing on using a different strategy to focus on recombination regions. Hereby I'll refer them as hotspots, where the parasite prefers to engage in mutation process and polymorphisms, and this will be important in identifying genome regions that are under selection and probably later on correlate with phenotypes and come up with potential drug targets that could be useful in therapeutic studies.
Salut, je m'appelle Ramata Mariko, étudiante de DELGEME de niveau master en bioinformatique à la faculté des sciences et des techniques de Bamako. Ma thèse de pharmacie était sur les caractéristiques biotiques et abiotiques des larves d'Anopheles gambiae sensu lacto. Ce travail a été fait au laboratoire de l'immunologie des vecteurs au Centre de recherche et de formation sur le paludisme. Mon projet d'étude est basé sur l'aspect bioinformatique et génomique des anticorps monoclonaux dans la prévention de l'infection des moustiques au cours des essais de gorgement direct sur membrane. Le but ultime de mon travail serait de répartir l'effet des anticorps monoclonaux sur la transmission des parasites du paludisme par les moustiques vecteurs.
Hi, my name is Desiree Nda Euni from Ivory Coast. I'm working at the Center for Diagnosis and Research on AIDS and other infectious disease in Trècheville at the University Hospital in Ivory Coast. I earned a DPharm degree from the Faculty of Pharmacy of Dakar in Senegal, studying the prevalence of cryptosporidiosis and mycosporidiosis to the Center for Diagnosis and Research on AIDS and other infectious disease for 10 years. I'm now a DELGEME master student at Jomo Kenyatta University of Agriculture and Technology in Kenya. For my master training, I will be working on genome-wide scan for Plasmodium falciparum diversity and drug resistance in Ivory Coast. My aim is to determine the susceptibility of Plasmodium falciparum to ACTs in relation to its genetic diversity in different epidemiological phases in Ivory Coast. The first DELGEME summer courses have been very useful for me, and I learned a lot of new things because I haven't a background in bioinformatics. So I hope we will be there in Bamako next year for more lectures to come.
Hi, my name is Elikwi Majo Amegashi from Ghana. I obtained a Bachelor of Science in Medical Laboratory Technology from the University of Cape Coast in Ghana. My final project was based on the anti-asthmatic property and possible mode of activity of an interlonic leaf extract of Polycius fruticosa. After I had a one-year internship at the 37 Military Hospital Central Laboratory in Ghana. I'm currently a DELGEME MSc student who will begin her MSc in Molecular Biology and Bioinformatics at the Jomo Kenyatta University of Agriculture and Technology this September. For my project, I seek to investigate the artemisinin resistance in Plasmodium falciparum populations in the Volta region of Ghana. This summer course has been very helpful. It has increased my scope of knowledge in bioinformatics.
Hi, I'm Sima Owenleo from Gabon. I need a master's degree in population and ecosystem biology at the University of Science and Technique of Masuku, Gabon, studying the ecology of mailbox in Ngamba or station in Gabon. Currently, I'm pursuing a master's degree in bioinformatics at the University of Science, Technique and Technology of Bamako, Mali. Under the DELGEME MSc student, I intend to work on the genetic variability and possible artemisinin resistance of Plasmodium falciparum in Gabon. The first course DELGEME summer course has been very useful for me, training and permitted me to get knowledge in different methods used in bioinformatics.
Hi, I'm Catherine Bakari from Tanzania. I'm working with National Institute of Medical Research, NIMRI, Tanga Center. I have a bachelor degree in molecular biology and biotechnology from University of Dar es Salaam, Tanzania. I'm now a master fellow under DELGEME. I'm expecting to start my studies at Jomo Kenyatta University of Agriculture and Technology in September. In my master training, tentatively I'll be working on Plasmodium falciparum histidine-rich protein 2 and 3 gene deletion and its effects on performance of histidine-rich protein 2 based rapid diagnostic test in Tanzania. My aim is to address the public health concern of misdiagnosis. Currently, I'm at the first DELGEME summer school and it has been very helpful for me, especially it has increased my knowledge and better understanding of bioinformatics.
Hi, I'm Musa Jimde from the Malaria Research and Training Center at the University of Science, Technique and Technology of Bamako in Mali. I did my medical doctorate degree at the Faculty of Medicine, Pharmacy and Dentistry at the University of Bamako. My thesis was based on the in vivo efficacy of 3 antimalarial drugs, namely Pancidar, Mefloquine, and Coartem, in the treatment of incubated malaria in children from 6 to 59 months old in Koro. I did 12 months training in biostatistics and data management at Competence Center in Methodology and Statistics at Luxembourg Institute of Health. It was under TDR WHO Capacity Building Fellowship. Actually, I'm doing a master's degree in bioinformatics at the University of Cape Town under DELGEME Fellowship. My dissertation project is based on the genetic characteristics of Plasmodium vivax from northern Mali, the surprising apparition of P. vivax in West Africa, and their ability to infect DAPI-negative black population is one more threat to population health. In order to contribute to malaria elimination effort, there is a need to investigate the genetic characteristics of P. vivax population and the divergence orthogous of gene conferring antimalarial drug resistance in P. falciparum. This is critical understanding the potential pattern resistance of the parasite to the drug in the northern of Mali. The aim objective of our project is to determine the genetic characteristics of Plasmodium vivax from northern Mali. So this first DELGEME summer course is a good opportunity for me to improve my skill in epidemiology, bioinformatics, scientific writing, and communication.
Hi, I am Javan Okendo from Kenya. I earned my bachelor's degree in molecular and cellular biology from Kenyatta University. I then worked as a research assistant at the Center for Research in Therapeutic Sciences, Strathmore University. I'm a DELGEME MSc student doing my Masters of Science in Bioinformatics at the University of Cape Town, South Africa. For my master's degree training, I will be working on the analysis of xenos evolution of Plasmodium falciparum during treatment with the aim of determining the presence of ACT drug resistance signatures. The first DELGEME summer school training has provided me with the prime opportunity to expand my knowledge base in bioinformatics and also networking opportunity with the young fellow African scientists.
Hi, I am Martha Nginya from Kenya. I earned my bachelor's degree in biochemistry from Pwani University, Kenya, and thereafter I did my internship at KEMRI, Center for Virus Research acute respiratory infection unit in Nairobi, where I was involved in surveillance of viral causes of respiratory infection. I then attained my postgraduate diploma in health research methods at Kemri Wellcome Trust in collaboration with Pwan University. My postgraduate diploma project involved analysis of genetic variation of apical membrane antigen 1 in Plasmodium falciparum isolate collected over 18 years in Kilifi County. Currently, I am an MSc DELGEME student at the Center of Biotechnology and Bioinformatics at Nairobi University, Kenya. I intend to work on Plasmodium falciparum histidine-rich protein 2 gene deletions. The first DELGEME summer school has greatly enlightened my bioinformatics knowledge, which will be important in my research career.
Hi, my name is Riba Abarra from Juma University Institute of Health, School of Medical Laboratory Science of Ethiopia. I'm earned BSc degree from Juma University School of Medical Laboratory Science of Ethiopia, studying medical laboratory science and pathology. I've been working in Juma University since 2014. Now I'm going to join Jomo Kenyatta University of Agriculture and Technology to take my MSc degree in bioinformatics. I would like to work my thesis on Plasmodium falciparum anti-malaria drug resistance. The DELGEME summer course training helped me to get valuable information about bioinformatics that increase my future performance. Thank you. I would like to acknowledge the DELGEME groups to give it such a golden chance. Thank you very much.
Hi, I'm Esokum Kevin from Cameroon. I earned my Bachelor of Science degree in biochemistry from the University of Buea in Cameroon. I went further to pursue my Masters of Science in Biochemistry in the same university. I'm currently a DELGEME Master student in Molecular Biology and Bioinformatics at the University of Jomo Kenyatta University of Agriculture and Technology in Kenya. I am interested in looking at variations in regions of ancient balancing selection in chromosome 4 of the human genome that encode important erythrocyte proteins that are involved in host protection against severe malaria in samples from Cameroon. The 6-week DELGEME summer course that I am currently attending is of great significance, as I've had the opportunity to meet with great men in the field of research in malaria and also young scientists in Africa. I am looking forward to many such courses to be well equipped for malaria elimination in Africa.
Hi, I'm Brenda McKenna from the University of Nairobi, Kenya. I have a bachelor's degree in biotechnology from Kenyatta University in Kenya. I also have a postgraduate diploma in health research methods from Pwan University in Kenya. I worked on analyzing the transcriptome of Plasmodium falciparum to identify differentially expressed genes in symptomatic and asymptomatic individuals. I am a DELGEME MSc student, currently doing my master's in bioinformatics at the Center for Biotechnology and Bioinformatics in Nairobi University in Kenya. I intend to do my research on assessing potential ACT resistance in regions of high and low endemicity of malaria in Kenya. The DELGEME summer course has served to broaden my knowledge on the field of bioinformatics and bridge the gap in my coursework.